Okay, listen carefully. I'm just going to complete Chapter 5, Part 2, which will still take a little bit of time. Chapter 6 and 7, I don't go anywhere near or into near the detail I do for my science major, so we should be able to complete those each day. You have an exam coming up in what, roughly a week and a half, a little more than that? I think it's the 13th, if I'm correct, or around there. Don't quote me on that, but it's in your schedule. So make sure you start studying this weekend if you haven't been. All right, we were talking about enzymes. Enzymes are catalytic proteins. What do I mean by catalytic proteins? They catalyze chemical reactions. Okay? We call it metabolism in a living organism. Metabolism is defined as chemical reactions catalyzed by enzymes in a living organism or a living cell. Now, when we talked about enzymes, we said they're proteins. Okay, and a protein's shape is directly related to its function. And we talked about how these proteins that catalyze chemical reactions, we give them a special name called enzymes, and that some of them need energy in the form of a molecule called ATP. Now, let me ask you all a few questions before we get going with this next part here, just to see where you're at, if anybody looked at the information. For enzymes, what is that energy-rich molecule that they usually need as a source of energy in order to help them do their work? What is that energy-rich molecule that they need? Right, ATP. Okay, so they need ATP. Now, ATP we talked about. There are two types of chemical reactions that have to do with making and breaking covalent bonds, except when the covalent bond is being made, that's the way the cell is storing energy. Okay, what do we call those types of chemical reactions? When the covalent bond is made, it's the way the cell is storing energy, therefore the product is rich in energy. What do we call that type of chemical reaction? Endergonic reaction. And then we said that energy-rich molecule that's produced through that endergonic reaction, such as ATP, when an enzyme needs energy, it'll grab onto that molecule and break that same covalent bond where energy has been stored in order to release energy so the enzyme can capture it and use it. What do we call that type of chemical reaction that results in the release of that energy from the energy-rich molecule? There's endergonic reactions, and what's the opposite of an endergonic, right? Exergonic reactions, okay. Now, we talked about enzymes also, and enzymes is protein, so a protein structure or shape is directly related to its function. But when we talk about enzymes catalyzing chemical reactions, I said enzymes, in order to catalyze a chemical reaction, they need reactants. However, at least one of those reactants, an enzyme will directly bind to, will directly grab to. What's the special name we give to that reactant that enzyme directly binds to? Right, the substrate. Now, what's the region of the enzyme that comes in direct contact with that substrate? What do we call that region of the enzyme? We don't say react site, but site's correct. What's the word that we actually use instead of react site? We call it what site? Right, active site. Okay. Wow, y'all did better than my major, so okay. Fingers crossed. All right, so what I just want to ask you a few questions. Some of this you'll need to know as we go through some for the rest of the information in this chapter. What I want to pick up with now is where we left off. Scientists, and especially biochemists, okay, bio means life. So when you take a biochem, or if you take a biochem course, you're focused on chemical reactions or chemistry of molecules in living organisms, whereas chemistry focuses on non-living and living. Biochem focuses on living. And scientists such as biochem, what they do is, not all the time, not 100% of the time, but most of the time, if a protein functions as an enzyme, they will give that enzyme a name that ends in ASC. Now, this is not 100% of the time, but it's very frequent, such as a pancreatic enzyme called DNase. Okay, the ACE person knows this is a protein that functions as an enzyme. This enzyme catalyzes a type of chemical reaction that involves hydrolysis. Okay, the prefix in its name, in this case, lets you know what the substrate will be, which is simply DNA. So this pancreatic enzyme released into your small intestines, DNase, okay, The DNA, or the first three letters, is actually, sometimes A is a capital. It's letting you know what reactant, or specifically what substrate it will bind to. The type of chemical reaction it will catalyze is a hydrolysis reaction. It will break the covalent bonds, linking these nucleotide monomers together, thus breaking DNA down into its basic building blocks, its basic DNA nucleotides, for easier absorption. So this is a digestive enzyme. But there's another type of digestive enzyme. Okay? It's called sucrase. Same pattern as with DNase. Ends in SE to let you know it's an enzyme. And then the first part of its name lets you know what substrate it binds to. Now keep in mind, okay, all substrates are reactants, but not all reactants are substrates. But it's in the name of the enzyme what it binds to. Now sometimes the prefix in the enzyme's name has to do with the product it produces, or it has to do with the shape of the substrate, not necessarily the type, such as sucrose. It might be the shape. Like there's an enzyme called Hexa Kinase. The hexo has to do with it binds to a molecule with 6 sides. Well, the molecule happens to be glucose. Okay. Kinase has to do with it adds a functional group called a phosphate group. So, but they usually, the name of the enzyme tells you what it does. Okay. In this case, this is a digestive enzyme that also performs a type of chemical reaction called hydrolysis. Its active site, right here, has a shape that complements the disaccharide sucrose. It'll bind to the disaccharide sucrose, and then the covalent bond holding the two monosaccharides, glucose to fructose, that make up sucrose. That one covalent bond will be broken through this type of chemical reaction. What will then happen is the enzyme will then release glucose and fructose, which are excellent sources of energy. Okay, so just to give you an idea of where they go, some of these names. In chapter 6, we're going to go over cellular respiration. Okay, there's a large group of enzymes, okay, that catalyze chemical reactions that make up this energy producing process called cellular respiration. But pretty much I just have you know one, and it's the star of the show. It's the enzyme responsible for synthesizing ATP through an endergonic reaction, and it's simply called ATP synthase. So sometimes the names are very simple and straightforward. Okay, now we're going to transition into our factors that affect enzyme activity. In other words, enzyme function. Listen carefully. These factors can either lead to activating an enzyme's activity, or they could enhance an enzyme activity. In other words, speed up the rate at which it's working. Or these factors could inhibit enzyme activity. Now listen carefully. Inhibit, okay, is actually defined 3 ways. Okay, the factors that inhibit enzyme activity either could inhibit enzyme activity by never allowing the enzyme to become active, or the enzyme could be active and the factor that inhibits the enzyme stops it from being active. It's already working, but now it's stopped. Or sometimes inhibit means the enzyme is active, but there's a factor that inhibits it and slows down the rate at which it's working, or the level of activity, okay? So these are the 6 factors: cofactors, chelators, temperature, pH, substrate concentration, the brackets mean concentration, and inhibitors. When we get to inhibitors, we're going to talk about actual molecules, 2 types, but they both prevent the enzyme from becoming active. Now the first 4, talk about them in pairs, because when you talk about cofactors, it's best to talk about chelators. They have opposite effects on enzyme activities, okay? One helps activate the enzyme. The other one removes this first one, thus inhibiting enzyme activity, so I pair them together. The next 2 factors I talk about together also, temperature and pH, because there are similarities between the 2, such as enzymes work best at a certain temp, called the optimal temp, and at a certain pH, called the optimal pH. Okay, so we'll go over those 2 together. We'll talk about the last 2 separate. So starting with cofactors, okay, normally what a textbook will tell you is it'll give you a fancy definition, a textbook definition. And I would tell students, look, I want you to define cofactors the way I give it to you. If you give me the definition in the textbook, I'm going to ask you to explain what it means in order to give you credit. And I've never had a student be able to do it. Oh, well, it's metal ions and blah, blah, blah. No, no. The definition I give you for cofactors is based on how I and others who worked in research labs applied this. Okay? Now, listen carefully. First of all, I'm giving you an example of 3 cofactors, so listen carefully. What I have in your notes isn't everything you need to know. Okay, it's just a fraction of what you need to know. Cofactors, the way I want you to know them, cofactors are cations. So don't know them as metal ions, even though that's not wrong. I want you to know cofactors as being cations, first of all. Okay? Cations that function as cofactors are bound by enzymes to help the enzyme fold completely into the correct shape. So cations that function as cofactors are bound by enzymes to help the enzyme fold completely into the correct shape. And then put a sub-bulletin under that to repeat what I've said multiple times. An enzyme shape is directly related to its function. Therefore, cofactors help activate an enzyme. Okay, or another way to say it is therefore cofactors help turn on enzyme activity. Well, if we met in person, okay, there's a lab, an enzyme lab you would have conducted, and you would have focused on 2 enzymes. One of the enzymes you find in your saliva, salivary amylase, but the other enzyme you find in fruits. The long name of the enzyme is Polyphenol Oxidase. It's the third part of its name where you have ASE. Okay? Polyphenol oxidase is the enzyme responsible for causing fruits to brown. Now, fruits, okay, produce seeds. So basically, any produce you buy that has a seed, it's a fruit. Okay? So polyphenol oxidase causes the fleshy tissue, the inner tissue of a fruit, to brown when exposed to the atmosphere. What they found is this enzyme needs 2 reactants. One of the reactants is found along with the enzyme in the tissue of the fruit. When you cut it open, the fruit, when you cut it open, that adds the second reactant, which is O2, atmospheric O2. However, what scientists is that in order for the enzyme to actually catalyze a chemical reaction using the 2 reactants, one of them being O2, to produce that brown coloring that you see in fruits such as avocados and bananas and things that we don't like, that brown coloring is actually, how do you say, an antimicrobial defense. It actually is supposed to interfere with microbes wanting to eat the fruit, although we see it as a fruit decaying. But what scientists discovered is that the enzyme PPO, it's not enough to give it the reactants it needs to produce the product, which has a brown color to it. They discovered that also in the tissue of the fruit are copper ions. Now it gives you 2, but I just want you to know these. So if you're not familiar with this, Cu is copper. These are copper ions. Okay? So what they found is that in order for this enzyme, polyphenol oxidase or PPO, to catalyze a chemical reaction that causes the tissue of the fruit to brown, okay, it's not enough to have the reactants. These ions have to be present, and they're showing you here these little red beads. These represent cofactors called copper ions. And what's happening, what you can't see, okay, is that this copper ions, it says water too, but these copper ions, I'm sorry, on each side, these copper ions are contributing to helping this enzyme fold completely into the correct shape so that its active site has the correct shape to catalyze a chemical reaction. Now, copper ions are not the only cations that function as cofactors, activating enzyme activity or helping the enzyme become active. There are also calcium ions and magnesium ions. Okay, so if we met in person, what you would end up doing is we would give you in solution the enzyme PPO plus its reactants, and also in solution would be the cofactor it needs, plus the two cofactors are all in the solution. And we would have you combine them, and then you would see the enzyme would cause the solution to turn brown, visually letting you know that the enzyme was active and it was producing a product. But then what we have you do towards the end of the lab, normally if it's in person, is we have you work with chemical reagents. These are solutions or chemical reagents called chelators. Okay, if you have the PDF doc, excuse the, or actually PDF or Word doc, I'm sorry, my grammar wasn't too good, but chelators remove cations from solution. Okay, therefore, chelators remove cofactors from solution. Okay, so it's twofold. Chelators remove cations from a solution. Therefore, chelators remove cofactors from a solution. Now, y'all tell me, how is that going to affect enzyme activity? Is it going to activate the enzyme? Is it going to enhance its activity? Or is the final result inhibition of enzyme activity? Yeah, so make sure you put that down. This results in the inhibition of enzyme activity. This results in the inhibition of enzyme activity. Okay. These are two types of chelators you would have used in the lab. Okay. One type of chelator is called EDTA, and they're chemical reagents. And when I say chemical reagents, I'm talking about they're in solution. They're chemicals in solution. It's called EDTA. But we would have given you a second chelator known as PTU. These are the full names, which I won't test you on, but I started having students asking me more frequently, what do the abbreviations or acronyms stand for? And I got the feeling at least one time somebody was being a bit of a smartass, to be blunt. So I started placing it on the PowerPoint, and I didn't get any more questions after that. Okay. Remember, in sciences, they love to give really long multi-part names to something, and then they use abbreviations. Okay. So if you look here, so what would have happened if we met in person is you would have had two test tubes. In both test tubes, you would have, in solution, added the enzyme PPO. In solution, you would have added the reactants it needed. In solution, you would have had a total of 3 ions you would have added to the solution, but it would have already been in the solution, so you technically didn't have to add this. All 3 cations or cofactors, copper ions, magnesium ions, calcium ions would have been in the solution. However, to one of the test tubes, you would have added PTU, and to the other test tube, you would have added EDTA. And what would have happened is the EDTA would have removed any magnesium and calcium ions in solution. But that's fine, because PPO doesn't need these cofactors. So when EDTA removes these from the solution, okay, you would have compared this tube with EDTA to your positive control, which would have had no chelators, and you would have noticed you had the same brown color. In other words, the chelator EDTA did not inhibit PPO's enzyme activity because it didn't need these cofactors. It didn't need these ions. However, in the other test tube, okay, where you would have added the PPO in solution, the reactants in solution, which would have also had all 3 ions, except now you add PTU, you would have seen a different result. You would have noticed the solution didn't brown because the PTU removes the copper ions from the solution. PPO has everything else it needs, everything, including temperature and pH. Everything else is ideal. But what happens is the PTU removes this cofactor, the copper ions. And what you would have seen in the tube is no color change. There would have been no brown color produced. That would have demonstrated how specifically the chelator EDTA, by removing these copper ions, inhibited PPO enzyme activity. Therefore, that also proves PPO needs what cofactor? It needs these. So sometimes the way a scientist proves an enzyme requires sometimes the way a scientist proves an enzyme requires a specific cofactor is by using chelators and removing them. By process of elimination, that's actually how they figure out what you need. Okay, you may have this yourself in everyday life. Okay, I can get by without driving, but if you take my phone from me, I'm going to be in a real foul mood, or vice versa. You ever had that happen? You figure out what you need by losing it? So that's kind of the way they work here. So when you study cofactors, put it together with chelators. Cofactors help activate enzymes, thus activate enzyme activity. Chelators do the opposite. By removing cofactors, they end up inhibiting enzyme activity. Now, temperature. Okay, I had to edit this PowerPoint because the initial PowerPoint they get said bacteria. But technically speaking, there are living organisms that live at temps way above mammalian body temp or way above human temp, 37 degrees Celsius. Some of these organisms live at temperatures approaching boiling temp of water. There's actually one species that lives above the boiling temp of water. Okay, we call them thermophiles or thermophilic archaeans. In other words, they're prokaryotic organisms classified under the domain archaea. Okay, they're a type of extremophile. They love extreme environments. So we're going to use these two. First of all, in regards to temperature, this is a general blanket statement. Enzymes require a certain temp that is their optimal temp in order to produce the amount of product needed by a cell. So enzymes require a certain temp, which is their optimal temp, in order to produce a certain amount of product required or needed by a cell. So the way it works in a cell, okay, is when we talk about an enzyme being active or enzyme activity, okay, for the cell, it's not about how much product can you produce, enzyme. For a cell, it's basically letting the enzyme know, this is how much product I need you to. I don't care if you can produce more. I don't need it. I need you to produce a certain amount so that we can function normally. Well, temperature's tied into this, okay? When scientists were looking at the temperatures of different living organisms and realized not all living organisms have the same, if you want to call it, body temp, even though these are unicellular, they're still individuals. The next question scientists asked was, okay, well, what is it about these organisms that results in them thriving and surviving and thriving better at these temps to where they need these temps? If they lose these body temps, it can have a negative impact on the living organism, could cost it its life. Well, what they discovered, one of the key reasons for organisms having to have a certain body temp had to do with enzyme function. Us, human beings or mammals in general, it's been discovered that one of the main reasons that it's important our body temp is 37 degrees Celsius has to do with enzymes produced by cells of our body. 37 degrees happens to be the optimal temp. But scientists, as technology improves, scientists came to discover there are living organisms, and granted they're unicellular prokaryotic organisms, they still function as individuals like you and I. And what they came to find out is that these living organisms lived at very high temps. And one of the reasons, if not the main reason why they lived at high temps, is because these high temps were the optimal temperatures for these organisms' enzymes to function properly, which in this case, it's the thermophile that lives at 77 degrees Celsius. Everybody, we're long gone dead before our body temp ever reaches 77 degrees Celsius. It's over with, okay? So listen carefully. They're showing you a curve here on these two. Now, if you notice here, the way it works, okay, if it's a human enzyme or if it's a thermophilic enzyme, it's the same pattern. If you expose the enzyme of either organism to temps below the optimal temp, the rate of reaction is the same thing as saying the level of enzyme activity. In other words, how active is the enzyme? That's what we mean by rate of reaction. How active is the enzyme? If you look, it doesn't matter whether it's a human enzyme or a thermophilic enzyme. It doesn't matter that their optimal temps are different. Notice the pattern's the same. If you start to decrease the temperature below the optimal temperature, enzyme activity gradually decreases. Okay, and eventually reaches zero, meaning the enzyme is no longer active. So in other words, when you expose an enzyme to temperatures below its optimal temp, that inhibits the enzyme, but it doesn't shut down the enzyme initially. It just slows the enzyme's activity or function down. However, look at the opposite side to the right. Doesn't matter what the optimal temp is, it's the same pattern. If you expose the enzyme to temperatures that are higher than its optimal temp, notice the enzymes are much more sensitive to temperatures that are too high versus too low. When you expose enzymes to temperatures that are too high, above the optimal temp, notice now instead of getting a gradual decrease, it's like jumping off a cliff. It almost drops straight down. Well, what they discovered is when you expose enzymes from different organisms to temperatures above their optimal temp, the reason why they more quickly become inactive, or the reason why their enzyme activity is more quickly inhibited to the point they stop functioning, is because high temperatures cause enzymes to denature. That means high temperatures cause enzymes to unfold. Therefore, let me ask y'all, when I say if an enzyme is exposed to temperatures that are too high above its optimal temp, if I say the enzyme denatures, okay, what am I saying about the enzyme shape? Is it losing its normal shape, or is it maintaining its normal shape? Yeah. And if it loses its shape, what else does it lose? If the enzyme loses its shape because it denatures, what else does it lose the ability to do? Function, right? So enzyme activity is inhibited. Okay, so very gradual slowdown in its activity when it's exposed to low temps below its optimal. But they're much, much more sensitive to temps that are too high. It'll cause them to denature and shut down. pH. Initially, the way we approach pH is the same way we approach temperatures, okay? Enzymes require a specific pH, which is its optimal pH, in order to produce the amount of product needed by the cell. So enzymes require a specific pH, which is its optimal pH, in order to produce the amount of product needed by the cell. Okay? Well, what they're showing you here are two enzymes. These are human enzymes, or more generally, mammalian enzymes. What else do these two enzymes have in common? They're classified under a larger category, they're classified under a broader category of enzymes called proteases. Proteases are enzymes that break down or digest other enzymes. Okay, so there's that ASE. Now, why am I telling you this? Because the name of these enzymes are pepsin and trypsin. Remember what I said, not all the time, but most of the time they'll give the enzyme a name that has ASE at the end. Not all the time. This is proof, not all the time. But both of these enzymes, pepsin and trypsin, they are digestive enzymes. Excuse me. Pepsin is a digestive enzyme produced by cells of the stomach. So sometimes it's called a stomach enzyme. Trypsin is a digestive enzyme that is actually produced by the pancreas and released into the small intestine. So it says intestinal enzyme, but it's not produced there. It's released into the small intestines. So that's where it does its job. That's why they call it an intestinal enzyme. They're both part of our digestive system. They're both released into our digestive tract. They're both involved in breaking down that portion of the nutrients you consumed that consist of proteins. Therefore, they're called proteases. They digest proteins, break them down, except pepsin was found to be produced by cells of the stomach because it needs an acidic pH in order to function optimally. Well, it functions roughly at about a pH of 2, rounded up a little bit. Okay. Well, what scientists discovered is not only do you have cells in the stomach that produce this enzyme pepsin, but you have cells in the stomach that produce the acid hydrochloric acid, or HCl. That HCl is responsible for this acidic environment in your stomach, pH of 2. The HCl helps contribute to the breakdown of nutrients, but at the same time, it's responsible for creating this pH acidic environment, which pepsin thrives in. It works best in. Okay? The problem is the content in your stomach, as it's being digested, eventually is moved into your intestines, specifically the small intestines. The small intestines, okay, what happens in the small intestines is that the pancreas releases digestive enzymes into the small intestines that don't like this acidic pH. They don't do well. Okay, they get all at this acidic pH. So the pancreas not only secretes or releases digestive enzymes into the small intestines, what they're not showing you is that the pancreas also releases a buffer into the small intestines, which causes the pH to increase to 8. Because pancreatic enzymes, okay, the optimal pH of pancreatic enzymes is a pH of 8, not a pH of 2. So as the nutrients you consume are moving through your digestive tract, from your mouth to your esophagus into your stomach, then they're exposed to an acidic environment, in part because there's an enzyme that works great at a pH of 2. But then as that content moves from your stomach into the initial first part of your small intestines, the pancreas has to adjust the pH and bring it up to 8, because the pancreas is also releasing digestive enzymes that don't work at pH of 2. They need a pH of 8, slightly basic. Okay. Well, an example of a pancreatic digestive enzyme is trypsin. Trypsin is also a protease. It continues the process that pepsin began, digesting proteins, but it can't work at a pH of 2. Notice here, as the pH decreases for this type of enzyme, it stops working before we reach a pH of 5. It works great at a pH of 8. So listen carefully though. It doesn't matter if we're talking pepsin, that requires an acidic pH as its optimal pH, or trypsin, which requires a slightly basic pH as its optimal pH. Notice the pattern when you decrease the pH. Unlike for temperature, where one side of the curve gradually decreases and the other side of the curve pretty much almost immediately bends downward, notice the curve on both sides for both enzymes is relatively the same. For both enzymes, if you decrease the pH, this slowly begins to inhibit the enzyme's activity. For both enzymes, it doesn't matter what their optimal pH is. If you increase the pH, you get the same pattern but in the opposite direction. Slowly, the enzyme activity is inhibited. Listen carefully. The way it works for enzymes in pH is that if the pH decreases too low or increases too high, the enzyme will denature. Okay, so again, for enzymes in pH, if the pH decreases too low or increases too high, this will denature the enzyme. And as we said before, if the enzyme denatures, it loses its shape, therefore it's inhibited. It's no longer active, can't function. Okay? So they kind of parallel each other a little, temperature and pH. There are differences, but there are some parallels between the two. Next is substrate concentration. Okay, I don't say a whole lot about this. Okay, I just basically use the graph to explain. Okay, the amount of product an enzyme can produce is dependent, okay, on the reactants, especially that reactant called a substrate, which the enzyme is actually grabbing onto and converting to the product. Okay, so an enzyme can have everything it needs: right temperature, right pH, the cofactor it needs, ideal water-based solution. Okay, but it still needs not only the reactants, but the reactant that is a substrate. So it can grab onto that reactant called a substrate and convert it to the product. So substrate concentration plays a role in how much product the enzyme can produce. But what scientists discovered is that enzymes can be saturated. In other words, they can reach a point where they're overworked. So you can continue to give them more and more substrate, and they're like, no, that's it, we're through. You may have had this happen at a job before. You're given some work to do, and you do it, and you're through quick. So they give you more work to do, but it's a larger quantity the next time, and you get it done. So then they give you more work to do, but you realize now the quantity is doubled, and you're starting to think they're taking advantage of me. But you get it done. So then they come back and they quadruple the quantity. And you realize you can give me all the more you want. I've already reached my peak. I can't work any faster than this. This is it. So throw all the more work you want at me, but I ain't going to do any more. I can only get so much done. That's the way it works for enzymes. So if you look here, reaction rate just means enzyme activity. Okay, the rate of enzyme activity. Now, down here is substrate concentration. They break the graph up into 3 boxes. First thing I want you to notice are these little guys here. They represent the enzyme. Notice from this box to this box to this box, the number of enzymes does not change. Okay, so when we talk about how substrate concentration also has an effect on enzyme activity, we have to assume the amount of enzyme doesn't change. The only thing that's changing is the amount of substrate. And that's what they're showing you here. So what they show you is initially, when just a little bit of substrate is provided to the enzyme, its rate of reaction is not very high. Okay, and the line here is representing product. You give me a little bit of substrate, I can only produce a little bit of product. In other words, I'm not very active because there's not a lot to do. But as you increase the amount of substrate or increase the substrate concentration, the enzyme says, okay, now I can work faster because there's more to work on. So I'm going to start producing more product. In other words, its enzyme activity begins to increase. Then we reach a point where it says here we have a medium amount of substrate concentration being given, okay, or more substrates being given. And the enzyme says, that's fine. I can keep up with this. Keep giving me more substrate, okay, give it to me faster. I can still convert it. I can increase my rate of activity, resulting in more product produced. But then when you get to the very edge of where it says medium substrate concentration, notice the curve flattens out. What this means is that the rate of enzyme activity has reached its peak. It's maxed out. The enzyme is saturated. It's saying at this point, right about here, on over, you can give me all the more substrate you want, but I'm not going to produce any more product than what I produced right before we reach this point. This is the maximum amount of product I can produce because that's the maximum amount of substrate I'm capable of converting to a product. Give me more substrate if you want. I can't work any faster. I'm saturated. I'm overworked. So that's basically what we mean. So give an enzyme too little substrate, it can't produce. It can't be very active because there's not a lot to work on. Therefore, it doesn't produce much product. Give an enzyme too much substrate, okay, and its level of activity is saturated. In other words, I can't work fast enough. I can't convert any more product, all right? So in other words, there's an optimal substrate concentration that an enzyme needs to produce the correct amount of product needed by a cell. So again, there's an optimal concentration of substrate that an enzyme needs to produce the amount of product needed by a cell. The last thing I want to go over is inhibitors. Okay. First of all, inhibitors are molecules. Okay. So when I'm talking about the 6 factors that affect enzyme activity and I get to number 6, inhibitors, this is what I mean by now we're talking about molecules, okay? You can talk about inhibitors and it could be a chelator, but what I'm saying is for number 6, the 6th factor that affects enzyme activity, I'm talking about actual molecules, not chelators, okay? Well, what scientists found is that there are inhibitors that exist in the living world that can inhibit enzyme activity or enzyme function. In other words, they can prevent the enzyme from ever becoming active. All right. And one of the two can actually turn it off, but we're going to approach it from preventing the enzyme from ever becoming active. Now, these molecules called inhibitors, however, they were found to not all be the same. In other words, their mechanism of action or the way they went about inhibiting an enzyme from becoming active wasn't always the same. Some of these inhibitors, the way they inhibited an enzyme from becoming active was by directly competing with the substrate for the enzyme's active site. Okay? These molecules, okay, they didn't have a shape that was 100% homologous or the same as the substrate. However, these molecules had a shape that allowed them to at least interact with part of the enzyme's active site. Interact with part of the active site strongly enough to hold on and block the enzyme from binding to its substrate. Thus, enzyme activity was inhibited. The enzyme never got a chance to be active because it couldn't even bind the substrate. Whenever an inhibitor directly competes with the substrate for the active site of an enzyme, inhibiting enzyme activity, we call that molecule not just an inhibitor, but we call it a competitive inhibitor. So what they're showing you here is a molecule, it's an inhibitor, but it's a competitive inhibitor because it's competing with the substrate for the active site of the enzyme. Okay. However, they found there were other molecules that functioned as inhibitors, except they did not compete with the substrate for the active site. Okay. What they found is there were molecules that could bind to an area on the enzyme away from the active site. Notice in this case, the inhibitor, the molecule, it's not competing with the substrate for the active site. Instead, it binds to a region of the enzyme away from the active site. But this inhibitor, when it binds to a region on the enzyme away from the active site, it causes the enzyme to lose its normal shape. And when the enzyme loses its normal shape, what else is lost? The shape of the active site. It no longer has the shape that complements the substrate. Therefore, the enzyme cannot bind to the substrate. Thus, this molecule has just inhibited the enzyme. It has prevented the enzyme from becoming active. Well, because this molecule is not competing with the substrate for the active site, but instead is binding to a region away from the active site, we call this type of inhibitor a non-competitive inhibitor. Okay? Are there any questions? Other than some of y'all might be thinking, God, I'm not a science major, and now I know why. Okay. Okay, listen carefully. I went back to the notes that I provided, the Word and the PDF doc, and I reshuffled the video links. Unfortunately, two of them are still to two of the video links are to the ACC Bio department, created by a couple of professors several years back. And it tells you when you first click on the link, it'll open up the internet and it'll say you need to click on Adobe Flash, whatever, in order to view the videos. So what I did is I put in a third video, and it's just titled Enzyme, and it's in the middle. Okay, so there's 3 videos now. I reshuffled them. But the second one that's titled Enzyme, and they all say watch this video, but the second one that says watch this video and it's titled Enzyme, that's a YouTube video from the Amoeba Sisters. So if you can't see the other 2, that middle one hopefully will be good enough to help you out. And it does go over some of the factors, okay? And I'll post the updated documents after we're through here.